People are more invested than ever in learning about their DNA. Companies like Ancestry.com and 23andMe have made personal DNA data increasingly accessible so that everything from forensics investigations, medical treatment, even Spotify playlists can be tailored to you as an individual based on the collection of DNA found in every cell in your body, your genome. As more people gain access to their personal genome, they're going to have questions. What does this mean? How does this stuff work? Right now, we don't really have complete answers. Because here's the thing. If you could actually pop open a living cell and take a look at a genome, what you would notice right away is that it's not just made of DNA. Alongside all of that DNA are thousands of proteins, each with a unique job. So if we want to understand how this genome works, we're going to need to know what these proteins are doing. So today, I'm going to tell you the story about one of these proteins, named Heterochromatin Protein 1, or HP1. And to do that, I want to bring your minds back to the 1958 Steve McQueen classic, The Blob. In The Blob, a mysterious alien organism crash lands and then continuously grows and expands, consuming everything in its path. This is exactly what HP1 does in your genome. It forms blobs that coat the DNA molecule and expand, swallowing up genes, blocking access to them so that other proteins can't swoop in and then turn those genes on by accident. So if you ask a scientist who studies the proteins packaged with your DNA, what does HP1 do? They'll tell you HP1 is a silencing protein. It turns genes off. But what if that's not quite the whole story? What if this whole time the blob has just been misunderstood? In my research, I've been looking at all the places in the genome where we can find HP1. One thing you'll notice right away is that HP1 is not just inside of the blob at genes that are turned off, it's also outside of the blob at genes that are turned on. What's weirder is that if you look at a special group of genes that only turn on in response to heat, you can find HP1 of these genes after they've been turned on, but not before. This doesn't make any sense if HP1 is just a blob that turns genes off, so maybe it's doing something else. In my thesis, I want to understand how HP1 can be the blob, but also be the exact opposite of the blob. How can it be involved in turning some genes off, but other genes on? Using fruit flies as a model, I'm studying HP1 in two ways. First, I take it out, and then I add it back in. I have a special group of flies that don't have any HP1. I can compare them to regular flies and see how their genomes are different. I also have a method to tie down HP1 to new sites in the genome and see what happens there. Does that new site turn on or off? Do any new proteins show up there? What are these proteins doing? In doing this, I'm revisiting the blob and painting a new picture of the genome so that when a doctor or a patient or even some guy at Spotify that's just trying to make a playlist have genomic data in front of them and are asking, what does this mean? How does this work? We're going to be able to give them a better answer.